Okay, iBio 30. I'm gonna finish up Mendelian genetics today. You have this practice question in your notes, so you can take a look at that. I'm gonna do one really similar to it in your textbook. This is a dihybrid cross. So I'm going to talk about dihybrid crosses today. That's what this is. So two traits. We've only been talking about one trait. So we're going to talk about two traits together. We're going to make Punnett squares based on 16 square Punnett squares and looking at two traits. So I'm just giving you an example here, just how to look at gametes with this first. So a couple things. So when we have two individuals, let's say there's gene A and gene B. So this is one individual in the breeding pair, and this is the other individual in the breeding pair. When you separate your gametes, you have to honor the law of segregation. So segregation is alleles that separate. Right, so these are alleles of the same trait. So alleles of the same trait have to separate without linkage, and then the genes, the different genes themselves. So that would be, so we're looking at alleles, that's segregation. And then the two different traits also have to separate in even numbers, and that would be independent assortment. So you also have to honor the law of dominance. So you always put dominant alleles before recessive alleles when you make your Punnett square. Okay, so just a bit of skill building with looking at Mendel right now. So when we make gametes for this, so this is individual 1, this is individual 2, we're going to separate this into gametes. So remember, if we just had one trait, you would have an A gamete, and then you would have a little a gamete. But now we have two traits together, so you actually use the FOIL technique. So first, this is one set of gametes, outer. There's your second set of gametes, inner. There's your third set of gametes, and then last. So that is the gamete pair that you'll make with this individual. Okay, and then you do the same thing here. So there's your first gamete, there's your second gamete pair, there's your third gamete pair, and there's your fourth gamete pair. Okay, so this is how we're going to make this Punnett square. Now these are both heterozygotes, so they have 4 possible gamete combinations, so 4 unique gamete combinations. So one individual goes across the top like this. And then we're going to put another one. Down the other side, and you use the same pattern. Okay, so when given 2 heterozygotes and a dihybrid cross, this is what you do. Now we're going to put the first, so when we make the gametes, you have to keep the first trait separate and the second traits separate from each other. Okay, so if I were to do this, that would be incorrect. Okay, so let's make this here. Big A, big B, little b. You always put it doesn't matter where the dominant allele is coming from, whether it's here or here, you always put it before the recessive allele. Right, so this would be A, little a, big B. This is A, big B, little b. So if you look at, so there's 1, 2, 3, 4 of these heterozygotes that go across the middle. You'll see that when you look at the gamete combinations. Okay. And then we fill the rest in. So big A, big B, little b. This is big A, big A, little b, big A, little b, little b. And this is big A, little a, big B, big B, big A, little a, little b, little b. This is actually little a, big B, and then little a, big B, little b. This is little a, big B, little b, and then there'll be one here that's little a, little b, little b. So if we have two heterozygotes for a dihybrid cross coming together, you're going to see that. This is a pattern that you need to pick out. So I'm actually going to show you a few things as well. So we don't really look at the genotype with dihybrid crosses, we look at the phenotype. So trait A, right, so we have a dominant phenotype here. I'm not really going to determine what the trait is, I'm just going to say it's dominant trait A, right? If something has little a, then it would have the recessive phenotype. And that would be the same with big B and little b. Okay? So what you'll see is when you have two heterozygotes, you're actually going to have one, right? So big A, big B, you're going to have 1, 2, 3, 4, 5, 6, 7, 8, 9 individuals in a 16 square Punnett square dihybrid cross that will have the dominant phenotype for both traits. So you'll have 9 big A, big B individuals. Okay, you're going to have 3 individuals that are dominant for the first trait and recessive for the second. So you'll see that here, here, and here. Okay. You will have 3 individuals that are recessive for the first trait and dominant for the second. Okay, and you will see that here. Okay. And then there will be one individual that will be homozygous recessive for both traits. So this is a 16 square Punnett square, and you'll always have this ratio when you see the offspring. So there'll be 1 little a, little b. So there's 16 squares. So the probability there should be 9 out of 16. You can convert that to a percentage that are dominant for both traits. 3 out of 16 which are dominant for the first, recessive for the second. 3 out of 16 that are recessive for the first, dominant for the second. And 1 out of 16 that should be recessive for both traits. Okay, so that's generally what you need to do when making dihybrid crosses, but you don't always necessarily need to have heterozygotes. So, for example, let's say you have 2 true breeders where they are homozygous for both traits, right? It's the same thing with a monohybrid cross. They're only going to make this gamete or that gamete, right? Because when you do FOIL, you only make big A, big B, and you only make little a, little b. So remember, it's not gross numbers, it's probability. So I can just cancel everything else out. And then when you make your Punnett square for that, you end up with offspring that have that. Okay. So let me give you a couple other combinations. Just need to erase my board. Okay, so true breeders could also look like this. So true breeders doesn't mean that one's completely homozygous dominant, the other's a completely homozygous recessive. You could also see this. These would also be true breeders. Okay? Those are also true breeders because they're homozygous for both traits. One's just dominant, one's recessive, but you make this and you make this. Right? So you still end up with, oops, the same thing. Okay? But you could also have, you know, let's say you have an individual like this and an individual like this. Okay? Where you have, you could have this gamete and this gamete and then this gamete that are made. So your Punnett square is based on what your alleles are. So you're not always going to make a 16 square Punnett square. It's redundant to do that. Okay, so you make your gametes based on the breeding pairs that are presented, not based on whether or not you should have a 16 square Punnett square. Okay, so just keep that in mind when you do Mendelian genetics or with dihybrid crosses. This is a test cross with Mendel, with dihybrid crosses. Right, that's the homozygous recessive condition. Okay, so let's do an example. So I'm going to do quite a few examples. So let's do a simple one. This says, in tomatoes, I'm going to pull out my notebook here again. So this is McGraw-Hill, page 596, number 4. So this says, in tomatoes, L. esculentum, red fruit is dominant to yellow fruit. So I'm going to write that down. So this is page 596, number 4. So red is dominant to yellow. Okay. And tall is dominant to short. So little t is short. So the question says, true breeding tall plants that produce red fruit, so you have tall and red plants, were crossed with true breeding short plants that produced yellow fruit. So this is asking you, asking you to look at a P generation, take an F1 and then turn it into an F2 with a dihybrid cross, rather than a monohybrid cross. So you've seen this before, you're just using it with a dihybrid cross now. So I have true breeding red and tall plants bred with true breeding yellow and short plants. Okay, so that's my P generation, and it's asking me to find my F1. So I'm going to have this gamete and this gamete. And I'm going to end up with, in the F1, 100% red and tall. Right? And they're all heterozygotes. Okay? My F2, I'm going to have two F1s bred together, right? So, when I do foil, right, I have 4 different unique gamete sets. So I'm going to end up with RT. Okay, so when I breed 2 F1s together, I have to do a 16 square Punnett square. And then when I fill this in, big R, big R, big T, big T, big R, big R. So this is what I get with two heterozygous F1s. So again, right, I'm going to have big R, big T, so there's going to be 9 of these. So 9 will be red and tall. 3 will be red and short. So I'm hoping you're starting to pick out these patterns. 3 will be red and short. 3 will be yellow and tall. And then 1 will be yellow and short. So I can apply numbers to this, right? So let's say there were, let's say there were 2,000 plants, right? So that would be 9 out of 16 times 2,000 would equal red and tall, right? 3 out of 16 times 2,000 would be red and short. 3 out of 16 times 2,000 would equal yellow and tall. And 1 out of 16 times 2,000 would equal yellow and short. Okay. And if I do that math, I will have 1,125 individuals that have they're dominant for both phenotypes. You should have about 375 individuals that are dominant for the first and recessive for the second, and recessive for the first and dominant for the second. And then you should have about 125 individuals that are straight homozygous recessive. Okay, so that's how you do just basic Mendelian genetics crosses. Number 5 is very similar on page 596. Okay. Page 598 is where I'm going to go next. So this, right, so there's quite a few good questions here, but this question, number 7, says, in fruit flies, long wings are dominant to short wings, and gray body color is dominant to black body color. If flies that are heterozygous for both traits are crossed, and 256 offspring are produced, how many of these offspring would you predict to have each phenotype below? So long and gray is the dominant trait, right? So you should expect 9 out of 16 to be big L, big G, 3 out of 16 to be this, 3 out of 16 to be that, and 1 out of 16 to be this. So if you put numbers to that, right, there's 256 individuals total. So you can do this Punnett square on your own. But I'm not going to do it because I already showed it to you. Okay, you should have about 144 individuals that are long gray. .48 individuals should be B and C. And then you should have 16 homozygous recessive individuals there. So I'm not going to draw that 16 Punnett square because I've showed it to you multiple times. It's the same thing. It's 2 heterozygotes. You just change the allele. So do that for practice. But 8 is a very good question. So 8 says, imagine that you randomly select one of the long-winged, gray-bodied flies from the offspring. And from the offspring described in question 7, and perform a test cross with a fly that is homozygous recessive for both traits. What is the probability that the offspring will be produced in the 1:1 to 1:1 ratio for long gray, long black, short gray, and short black? So 25%, all of these different phenotypes. Okay? So you're going to So this is another common trend or style of question that you see with dihybrid crosses. Okay, so again, if I'm actually going to draw this. So here's the offspring. Okay, and then when we fill this in, so by now you should be able to start to pick these patterns out, right? Okay, so the question is asking if one of these offspring is crossed with a test cross. So I'm going to take one of these offspring and I'm going to cross it with a test cross. Which one gives you this 1 big G LG to 1 big G little g to 1 little g big G to 1 little L little g? Okay, so which one of these would give that to you? So this is trial and error, but I'm going to point out that the only genotype that's going to give that to you when you cross with a test cross, you're going to cross a heterozygote for both traits with a test cross to get this, all right? So this 1 to 1 to 1 to 1 ratio is always going to be a heterozygote with a test cross. So when we make this Punnett square, right, it's little l little g, right? And then you're going to have big L, big G, right? Big L, little g, little L, big G. And so here's my gametes. From that, and then when I cross with the test cross, so that's the only, so this should be, when you think about it, it should be kind of intuitive because the heterozygote is the only one that gives you 4 gene combinations and you're looking for a ratio that's divided into quarters. So what you get when you cross a heterozygote with a test cross is you're going to get 1 long gray, so that's long gray, right, to 1 long black, and then you're going to get 1 short gray, and then to 1 short black. Okay, so this is this heterozygote will be the one that gives you that 1 to 1 to 1 to 1 to 1 ratio when you breed it with a test cross. So how many of them are there? There's 4 out of 16 that are big L, little l. So the probability that you'll get this ratio when you breed one of these F2 offspring All right, with a test cross is 25%. All right. Okay. So page 598 is very good, McGraw-Hill, a very good piece there. Do some practice questions with. I'm going to go to your Nelson textbook. And we're going to look at a few more questions here. Okay, so page 619, also very good. Okay, question 1 is more of a straightforward. This is Nelson now. This is a more straightforward question. But I'm going to look at question number 2 here. So this is Nelson, page 619. We're going to do a few of these questions, actually. Question number 2. So the question says, black coat color in Cocker Spaniels is, so I'm going to write this down. Black is dominant to white. Solid is dominant to spotted, so little s is spotted. Okay, the gene for pattern arrangement is located on a different chromosome than the one for color, and the pattern gene segregates independently of the color gene. A male that is black with a solid, so this is, again, this is like another forensics question. Okay, so I'm going to write this stuff down. So a male, so the male is black, so your male here is black, and he has a solid pattern. So he's at least has one big B and one big S. And he mates with 3 females. So he has female A, female B, and female C. So the mating with female A, so female A is this. So if I look at female A, she's white. So she has to be little b, and she is a solid pattern, so she has at least one. She has at least one S chromosome. So it's asking you to indicate the genotypes of the parent. So from the offspring, we got to figure out what the male's genotype is, what female A, B, and C is. Okay, so they produce 4 pups. So there's 2 black and solid pups. So there's 2 of them. And then there's 2 that end up like this, white and solid. Okay. So here's the deal. In order to get 2 little b alleles from, in order for some of her pups to be little b, then this male has to have at least 1 little b allele. So we know that he's big B, little b. Okay. Not quite sure about the solid coloration yet, though. All right. Because if they're heterozygotes, you could still get 2 big S alleles from the parents. So let's look at the next one, female B. So the mating with female B, which is black and solid. So this female is BS. Okay. This produces a white and spotted pup. So the pup is actually this, homozygous recessive. So this tells us right now that the female is a heterozygote and the male has to be a heterozygote. Okay. Because in order to get two little S alleles, one has to come from each parent. And then in order to get the two little B alleles, one has to come from each parent. But they're both dominant. So we're not really sure about this one. I don't think we're really going to know that one. Okay. The mating with female C. So female C is white and spotted. So female C, she is little b, little s. And we're done. Okay. So she's a test cross. And then you get a 1-to-1-to-1-to-1 ratio of whatever's there. Okay. So that's how you'd solve that question. Let's look at number 3. Now, one thing you should understand is that dominant and recessive genotypes, they don't necessarily have to be letters, and that's what this shows. Okay. So it says for human blood, the alleles for types A and B are codominant, but both are dominant over the type O allele. The rhesus factor, Rh factor, you guys talked about that in Bio 20, is separate from the ABO blood group and is located on a separate chromosome. The Rh plus allele is dominant to Rh negative. Okay, so A, right, is IA, B is IB, and O is I. Okay, but plus is rhesus factor and negative is Rh negative. Okay, so I don't need to, I can just put plus and negative, I don't need to put Rh plus, Rh negative. So these act just like, it's a dihybrid cross, but the alleles look a little bit different. So what this says is indicate the possible phenotypes of a child of a woman with type O blood, and she's Rh negative. So the woman is II negative. And a man with type A Rh plus. So we're going to make him a heterozygote because you're going to get more allele combinations. It doesn't say if he's dominant or recessive, so you just assume he's heterozygous. And this is plus negative. So what are the phenotype combinations of children? Make your Punnett square. So there's only one possible gamete there, I negative. And from this one I get IA plus, IA negative, little I plus, and little I negative. Okay? And then we make a Punnett square just like we would with anything else.